Hello everyone. So now we will visit the waters of Marseille a little bit. Today I am pleased to be here to present to you a part of my postdoc on the usage of epiphyte assemblages present on the leaves of Posidonia oceanica in the sea as bioindicators of contamination, more specifically metallic pollution today. So let's go to the Mediterranean Sea, and here we can find that the Mediterranean Sea is a hotspot for a lot of biodiversity. But unfortunately, in the context of multiple stressors, we can find a lot of stress present in this ecosystem. And we can cite mainly chemical pollution like metallic pollution, or either pollutants like persistent organic pollutants, and many other substances that can create dangerous mixtures. And another problem is the water temperature variation, which can be related to climate change. So here, basically, I show you a seagrass of Posidonia oceanica. And in these pictures, we can see on the leaves epiphytes. So basically, those are microorganisms present on the surface of the leaves. And currently, Posidonia oceanica, which is a protected species, is vulnerable to anthropogenic activities. So today, in my project, we are asking ourselves the question, how metals can influence the structure and the composition of these epiphytes. So in this presentation, first I will present the biodiversity of epiphyte present on the leaves. And then I will show you a little bit metallic pollution measured in these leaves. And finally, I will present an example of how we can link metallic pollution to epiphyte biodiversity and maybe propose it as a bioindicator. So basically what have been performed is samplings in 17 sites from Fos-sur-Mer until Pradé and also in Corsica. So we have chosen sites close to Posidonia oceanica. And these sites are different in terms of anthropogenic pressures, so basically urbanization, industrialization, ports, and wastewater treatment plants. And we've monitored those sites from 2017 to 2021. So after diving underwater and collecting the leaves, in the laboratory, we have extracted the epiphyte assemblages on the leaves. And afterwards, it was followed with freeze-drying, crushing, and extraction of eDNA. The method we use is metabarcoding. And what is interesting here is that we focus on two universal genetic markers, which are CO1 and 16S. And afterwards, so we've got, since we're working on microorganisms that we cannot always see, we've got the sequences present in each sample. And with the bioinformatic techniques, we can match our sequences with the available sequences of certain species or taxa. And eventually, we obtain which epiphytes are present in these samples. So concerning the first part on those biodiversity of epiphytes, here I show you a first result, general result. So for the 16S dataset and for the CO1 dataset, what we can see here is the number of taxon based on different phylums. So basically, we've got in all our sites dominantly proteobacteria, actinobacteria, and cyanobacteria. While in the CO1 dataset that is focused on metazoans and algae, here we can see first for metazoans that we've got a lot of arthropoda, cnidaria, and bryozoa. And for the algae, we've got a lot of rhodophyta and phaeophysae. So now we will dive in more details. And so I chose the example of the site of Fos-sur-Mer because we've got 17 sites. We're not going to present all of it in 10 minutes. And Fos-sur-Mer is a bit well known for its industrialization. So basically here in the 16S, what we see in this multivariate analysis is the distribution of assemblages of epiphyte based on different seasons. And when they are close on this figure, it means that they are correlated positively. So if you look more closely, for example, we've obtained the results in summer 2018, summer 2019, summer 2020, and what we can see on those bar plots is the composition of those epiphytes. So in summer, we've got a biodiversity composition that is slightly different between summers. So we've got some colors that are conserved and some others that can change. So basically, those are species or taxa that can change. We've done the same thing in autumn 2019 and winter 2019. And what we see is that the composition of epiphytes are different from the ones in summer. And then, so for the CO1 metazoan data set, it's the same approach. It's a bit more complicated. We've got more data. It's a bit more heterogeneous. And what we can see is that in winter, spring 2018, 2019, we've got the biodiversity composition that is a bit different between the seasons of different years. And we've obtained the results in summer 2017, 18, 19, and 2020. And here we've got differences in biodiversity composition. And finally, for autumn, the composition of biodiversity is also slightly different. So basically, over the years and over the seasons, for the CO1 metazoan, we've got biodiversity composition changes in epiphyte. And finally, for the algae, here we've got winter 2018 and 2019. So the yellow is the same and the green species, but we've got some differences. And in summer 2017, 18, and 19, we've got a bit of conservation of species with slight differences. And here we've got autumn. So overall, biodiversity changes between seasons. We can discuss more details later. And afterwards, what we have done is also another type of experiment is to measure the metals in the leaves with mineralization and ICP-MS method. What we obtain here, so we've got a list of metals that have been measured and in the leaves, in the epiphytes actually. And what we see is correlations on this PCA. We see correlations between metals. And on the other part of the PCA, we've got the sites. So for instance, if we take the blue representation on the right, up, it's for the site of Samena. And here we can see that it's correlated positively with some metals that are also in here, like zinc or lead. And if we take the other example of phosphore-mer, that is in green on the right, it's correlated to other types of pollutants, like, for example, aluminium or manganese. And the third part of our experiment is to link these metals with the biodiversity and to see if, for instance, certain biodiversity taxa can be indicators of certain types of pollutants. And here in this multivariate analysis, that is a bit different, but also we've got the positive and negative correlations. What we can see is basically the metals on the arrows, and the numbers are the taxa numbers. And here, without seeing all the detail, we can see that a lot of taxa can be correlated positively with different types of pollutants, and negatively also. So basically, we might have some indicators of sensibility or tolerance of substances. The same approach was applied also to CO1 metazoan, and we've obtained also a list of indicators, taxonomic indicators, and also algae. So finally, concerning our project, we are currently applying the same methodology to other types of substances that are problematic, like persistent organic pollutants, and more specifically flame retardants. And what we are also trying to study is the impact of temperature variation over the years, to see also if it influences in the same way as metals, or maybe it could be cumulative. So those are the perspectives of our project. Thank you for your attention, and I would be pleased to answer all of your questions.